Hello everyone. Today we are going to talk about the second topic in the series, G-protein coupled receptors. So this is on our series of biochemical signaling. So last time we talked about hormones and other nonpolar molecules that could get into the cell just by diffusing through it because of their nonpolarity. Today we want to start with the heterotrimeric G proteins, and this is covered in sections A through D in 13.3 of our textbook. So first let's talk about the receptor. So we're going to talk about each of these proteins in order of how they are activated. So the G protein coupled receptors, this is the largest family of cell surface receptors, and about half of all known drugs work through these receptors. They're called G proteins because they bind GTP, and there are different classes of G proteins. And the one that we're probably most familiar with is what's called G alpha sub S. This is associated with adenylyl cyclase, and the second messenger is cyclic AMP. So we would have talked about this in earlier classes using epinephrine and glucagon as signaling molecules. So these G proteins are linked to receptors that have similar structures. So even though there are quite a few signaling molecules that bind to the receptors, these receptors themselves have a similar structure. So let's look at that structure. So this is the receptor itself. So it is a multi-pass membrane protein. It passes through the membrane 7 times, and H here just means helix. So we have the amino terminal in the cytosol, and then it passes through the membrane 7 times so that the carboxyl terminal end also ends up in the cytosol. This big loop right here is where the receptor interacts with the G protein. So each of these cylinders is an alpha helix, and that's shown up here up at the top in sort of a rainbow order of what this receptor looks like. So again, this is a membrane receptor, and there's a large domain right here that interacts with these various G proteins that I showed you in the last slide. So here's the G protein. So this is a heterotrimeric G protein. So it has 3 subunits, alpha, beta, and gamma. And here's sort of a top view over here on the left. And this is the view of the G protein interacting with its receptor. So this is the beta-2 adrenergic receptor. So the ligand here would be epinephrine. And again, here's the receptor in its rainbow order, passing through the membrane 7 times. And then this is the portion of the receptor that interacts with the G protein. So we see we have an alpha subunit, a beta subunit, and a gamma subunit, so that the trimeric G protein interacts with the receptor on the cytosolic side of the membrane. The alpha subunit contains the GTPase activity, so the G protein is inactive when all 3 subunits are associated with one another and when GDP is bound. So activating the protein means that alpha dissociates from beta and gamma, and GDP is swapped for GTP. So this is shown in this sort of cartoony figure right here. So when the ligand, the extracellular ligand binds to the receptor, it causes a conformational change that activates the G protein. So the G protein is shown here associating with the receptor. They probably should have made it the same color. It makes it sound like look like the same protein, which it's not. So anyway, so this is the alpha, beta, and gamma subunits. They are anchored into the membrane by fatty acyl chains that resemble the fatty acyl chains of the phospholipids that make up the membrane. So the extracellular ligand binds, activates the receptor, and then when the G protein is activated, it swaps GDP for GTP, and it dissociates from the beta and the gamma subunits. So what happens after the G protein is activated? So the G protein acts as a transducer of the signal. So when it is activated, it activates some kind of a target protein. So the common target proteins include adenylate cyclase. So we want to talk about that first. It can also activate phospholipase C beta. We'll talk about that at the end. Or it may open an ion channel. So there are lots of proteins that the G protein might activate. Again, it might activate enzymes, but it might open channels as well. It depends on what type of G protein is associated with that receptor. So let's talk about adenylyl cyclase or adenylate cyclase. You find the two different names in the literature. So here's ATP, and the adenylate cyclase catalyzes the formation of cyclic AMP. So cyclic AMP is the second messenger because the first messenger, the hormone, never gets inside. So we make the cyclic AMP, and these two enzymes sort of are the balance of these two enzymes will determine the concentration of cyclic AMP in the cell. So if we have high adenylate cyclase activity, then the concentration of cyclic AMP in the cell will be high. But the cyclic AMP phosphodiesterase activity will hydrolyze the cyclic AMP to form just regular AMP, which is not a second messenger. So the balance of the activities of these two enzymes are important to maintaining a certain concentration of cyclic AMP. So here is the association of all three proteins that I just told you about. Okay, so here's the receptor again. Remember, it's that 7 membrane pass protein. It's not shown that way in the drawing, but the hormone binds, and then here's the G protein. And again, here it's not shown that there are 3 subunits, but there are. It's anchored in the membrane, and we can see it's inactive because GDP is bound. So when the hormone binds, and again, the dissociation of the alpha, beta, and gamma subunits isn't shown here, but the activation of the G protein occurs when GDP is swapped for GTP, and the alpha subunit dissociates from beta and gamma. So the activated G proteins, that's shown here with the GTP in green, activates the adenylate cyclase, so that's shown in this gray blob right here. So that enzyme converts cyclic AMP to, ATP to cyclic AMP, and then cyclic AMP brings about the cellular response that's the directive of the hormone. The G protein is eventually inactivated by the endogenous ATPase activity that you find in the alpha subunit. So the GTP is hydrolyzed to GDP, and then the signal turns off. So if you want to keep the signal on, you have to keep having the hormone bind to the receptor. So let's look at the structure of the adenylate cyclase, since it's not shown in any detail in any other slides that I showed you. So it is a membrane-anchored protein, but it has this large cytosolic domain, and that is where the active site is located, where ATP is converted to cyclic AMP. And of course, there is a domain, another cytosolic domain on the enzyme that associates with the G protein, because it's the association with the active G protein that activates this enzyme. So now what happens? So I find this very interesting that what's happened now is that we have a protein that's bound to the receptor, activated the G protein, activated adenylate cyclase, and now we have a high concentration of cyclic AMP in the cell. So what does cyclic AMP do? Well, it depends on what cell it's in, and if you look at this table, it is quite interesting that depending on what tissue you're in, so that's shown in this column right here, and this shows the variety of hormones that are binding to the receptors, and these are the cellular responses that you get depending on the hormone that binds and depending on the cell that you're in. So sometimes cyclic AMP opens ion channels, but most of the time it activates protein kinase A. And again, what protein kinase A does is it phosphorylates other proteins, but what proteins get phosphorylated depends on what cell you're in. So let's look at protein kinase A. So what happens is, this is what the structure of cyclic protein kinase A looks like. It has 2 catalytic subunits shown here in blue, labeled C, and then this is a regulatory subunit, and you can see that the regulatory subunit is binding to the substrate binding clutch, so that the substrate can't bind. So what you need is 4 molecules of cyclic AMP to bind. And when it binds over here to the regulatory subunits, and that causes a huge conformational change that frees 2 copies of the active protein kinase A. And now the protein kinase A is able to phosphorylate cellular target proteins. This is from the protein data bank. It was the molecule of the month a while ago. And there's a picture here from Susan Taylor's group. Her group has really defined protein kinase A and learned so much about it. So in this picture over here on the left, the catalytic subunits are shown in pink, and then this shows the conformation of the regulatory subunits. And then when cyclic AMP binds, so cyclic AMP is a sort of this fuchsia color right here, the conformational change occurs that frees 2 copies of the active protein kinase A. So over on the right, we have even more detail, and this shows these flexible linkers of the regulatory binding subunit, and how you can see this helix part right here and how it blocks the catalytic binding site. And down here, you can see the conformational change from when cyclic AMP is bound and when it's not. So the green is the conformation the active site has when cyclic AMP is bound. So that would be the active form of the enzyme. And you notice it's not, the conformation here isn't really that much different, but it's different enough to make a difference of the activity. But of course, we have the regulatory subunits blocking the active site completely. So those are removed when cyclic AMP binds, and you can get this huge conformational change that releases the catalytic subunits. So here is a structure of what the catalytic subunit of protein kinase A looks like. So this is from our textbook. So this shows a ribbon diagram, and the substrate is just shown in this orange ribbon right here. So remember, substrates of protein kinase A are other proteins. So here, this is just a little protein piece right here, a 20 amino acid peptide. And in white right here, that is an alanine. So that would have been either a serine or threonine that would phosphorylate, that would be phosphorylated by protein kinase A. And here is ATP. So you notice how ATP is very close to this residue right here, which it would have to be, because that's the phosphate right here that is donated to, if that were a serine or a threonine, would be donated to that protein to phosphorylate it. In stick form, it is shown the catalytic amino acid side chains. Remember, we've talked about this before, how X-ray crystallographers will often have an inhibitor in the active site just so that you can identify the active site, but you want to keep the enzyme still so that you can take a picture of it. But this is what it looks like. So what we do is we take an ATP and we use that as a phosphate donor, and we phosphorylate either a serine or a threonine on a protein target. So I want to remind you that phosphorylation is a molecular switch. So gaining or losing a phosphate group determines whether a protein is active or inactive. So what's difficult is that sometimes proteins are activated by phosphorylation, and sometimes phosphorylation inactivates them. So we just have to think about the context of what we're talking about so we can figure out which is which. So a kinase throws the switch in one direction by phosphorylating the protein using ATP as the phosphate donor, which is shown in the last slide. And again, phosphorylation makes some proteins active and other proteins inactive. And these are serine threonine kinases. And a phosphatase throws the switch in the other direction by removing the phosphate group by hydrolysis. So these are serine threonine kinases. Tyrosine kinases are in another class, which we'll talk about later. So this is just a picture to show what we mean here. So here the substrate protein is shown phosphorylated and active because the kinase has phosphorylated it using the ATP as a phosphate donor. And in the other direction, the protein phosphatase has catalyzed the hydrolysis of the phosphate to remove the phosphate. And now we just have the side chain of either the serine or the threonine. So this picture shows the phosphorylation. It shows how you switch it, but I could just as easily change this figure to write inactive up here and active down here, because it depends on the protein. For some proteins, phosphorylation is an on switch, and sometimes it's an off switch. Okay, so let's add to our picture right here of all of these players. Our signaling molecule binds to the G protein coupled receptor, and it activates the G protein, which is shown inexplicably over here for some reason. The G protein swaps its GDP for GTP and dissociates from beta and gamma, and then this active form of the G protein activates adenylate cyclase, which is shown here in brown. And then the adenylate cyclase uses ATP as a substrate to make cyclic AMP. And then cyclic AMP binds to the regulatory subunits of protein kinase A. And now we have two subunits of protein kinase A. So there are lots of things that protein kinase A can do. If you remember in biochemistry 1, we talked about the role of protein kinase A in glycogen metabolism in the liver. Um, but G protein coupled receptors do a lot of things. So the protein we were talking about now is the G alpha sub S. So this is a protein that is the alpha refers to the alpha subunit and S is stimulatory. So that means that when the G protein is activated, the adenylate cyclase is stimulated and the concentration of cyclic AMP is increased. It is possible, though, to have a G alpha sub I where the adenylyl cyclase is inhibited instead of activated, in which case cyclic AMP concentration will go down. So this is G alpha sub I. So there are different cells, different receptors that work via G alpha sub I rather than G alpha sub S. So there's an animation on the Wiley website that is the link here is live and it's posted on Sakai, so I'll let you watch that. But we've been talking about G alpha sub S, where we eventually end up with a high concentration of cyclic AMP that will activate protein kinase A, and then the protein kinase A will phosphorylate certain proteins, and whichever proteins are phosphorylated lead to a certain response. Then this is the inhibitory signal. So other cells work this way, where the activation of the G protein actually inhibits. So there's a nice little red arrow here, right here, that inhibits the adenylate cyclase, and that would cause the cyclic AMP concentration to go down. So for those particular cells, a decrease in cyclic AMP has different cellular effects. So we've put into motion this signaling pathway, but we need to throw the switch in the other direction. If we're going to use glycogen metabolism as an example, an organism will switch from the fed state to the fasted state or fasted to fed state. So we need to be able to turn on the pathway, but we need to be able to turn it off when the signaling event is over. So there are lots of phosphatases. There are 4 classes of phosphatases that remove phosphate groups by hydrolysis. So protein phosphatase 1 removes many of the phosphate groups and proteins that were phosphorylated by protein kinase A. So there's an animation on my YouTube channel. I posted it there, so you can go and look at that. But there are other ways that the cell has to make sure that if the cell is exposed to a high concentration of a signaling ligand for a prolonged period of time, the cell has ways of turning off the signal, and that's called desensitization. So one of the things that the cell could do is that maybe it could endocytose the receptor. There are G protein coupled receptor kinases that can phosphorylate the receptor, and then that could be followed by a binding of a protein called arrestin, and arrestin recognizes the phosphorylated receptor. So even the cell would be turned off if the signaling molecule concentration went down and failed to bind. But there are ways to turn off the signal even if for some reason the signaling molecule bound for a long time. So this is sort of the cell's response to like, well, maybe something is wrong if the signal is on for this long of a period of time. So I wanted to remind you of the system that we learned about in Biochemistry 1, and some of you were with me in nutrition. So here's protein kinase A up here at the top, and it's active. And protein kinase A has various cellular targets. So this is increased cyclic AMP. So this is what would be happening in the liver if you were fasting. So the hormone binding to the receptor and activating the G protein, which activates adenylate cyclase, would have been glucagon, because that's the hormone of the fasted state. So there's an enzyme called, this textbook author calls it glycogen phosphorylase kinase. I think I just called it phosphorylase kinase, but that enzyme is phosphorylated. It's a kinase, so then it goes ahead and phosphorylates glycogen phosphorylase. And remember, we learned that glycogen phosphorylase is the enzyme that carries out the process of phosphorylasis of glycogen at the non-reducing end to release glucose 1-phosphate. And if this is the liver, and it is because we're fasting and it's glucagon, that phosphate group is removed, and the well, first you convert glucose 1-phosphate to glucose 6-phosphate, then glucose 6-phosphatase would remove that phosphate, and then glucose could leave the cell for the benefit of other body tissues during starvation. At the same time, if protein kinase A is stimulating glycogen breakdown, we want to make sure that we inhibit synthesis. So another target of protein kinase A is glycogen synthase. So if glycogen synthase is phosphorylated, phosphorylation makes glycogen synthase inactive. So this is an example where over here on the left, phosphorylation makes the phosphorylase kinase and the glycogen phosphorylase. That activates them. I would have colored this in green if I had been making this figure. But here, the glycogen synthase is turned off. The other thing we want to turn off is there's this inhibitor protein right here, and protein kinase A phosphorylates it as well. And we remember we have that protein phosphatase 1, and we want to make sure that if we're turning on the signal by phosphorylating these 3 proteins, we want to make sure that protein phosphatase 1 doesn't undo our good work by removing the phosphate. So this inhibitor protein, when it is phosphorylated, tethers the protein phosphatase 1 and inactivates this. So what we have here is in the liver, fasted state, protein kinase A has phosphorylated 4 targets, and the end result is the breakdown of glycogen and the inhibition of its synthesis. And in this way, glucose is released from glycogen breakdown to the other body tissues that need it during starvation. So what happens if when you're starving and then you eat again? So in this case, the concentration of cyclic AMP would be decreased because glucagon wouldn't be made anymore. And that means you wouldn't be activating adenylate cyclase, and then that would give cyclic AMP phosphodiesterase a chance to hydrolyze it all to AMP. So now, we talked about this in nutrition about how insulin activates protein phosphatase 1. So that now is going to remove the phosphates from all of those targets. So now the glycogen phosphorylase kinase and the glycogen phosphorylase are now inactive, so glycogen won't be broken down anymore. We remove the phosphate from glycogen synthase, and now this is active. So this should be green. So this again, now the glycogen synthase will take UDP glucose and synthesize glycogen, because that's what we do when we're fed. We have the resources now to be able to use glucose from the diet to store it in the form of glycogen for the next time that we need glycogen breakdown during a period of starvation. So what happens after the G protein is activated? So I talked about adenylate cyclase being activated, but there's also some cells that use phospholipase beta. And this is a different class of G protein. So this is a G protein called G sub Q. So let's talk about those. So those are probably unfamiliar to us. So that means that we've progressed to section 13.4, which talks about the phosphoinositide pathway. So let's talk about that. So here again is my slide with all of my G protein, different kinds of G proteins. So we talked about G alpha sub S and briefly G alpha sub I. So this is G alpha sub Q. So the associated vector here is not adenylate cyclase, it's phospholipase C. And the second messenger here, we have 2 of them. We have IP3 and diacylglycerol. So there are different receptors. So this would be an alpha 1 adrenergic receptor, for example. So let's talk about how this pathway works. So what we have to do is we have to address another concept called calcium. So in the cytosol, the calcium is very, very low. And the cell maintains these low concentrations, say 10 to the minus 7th molar, in the face of very high extracellular calcium. But the other thing, we have calcium sequestered in the cell. The endoplasmic reticulum also has a high concentration of calcium. So the cell has ways of using calcium. When calcium floods the cytosol, that's a signaling event. And then after the signaling event is over, then you have to pump the calcium back in to its various places. So this is how the cell normally maintains these low concentration of calcium ions. We have a sodium-driven calcium exchanger where sodium comes in and calcium leaves. Of course, we're going to have to then eject the sodium using the sodium-potassium ATPase pump, which is not shown here, and that would be an example of secondary active transport. We do have primary active transport too, where calcium leaves the cell. And again, since this is against a concentration gradient, we need to have the phosphorylation of the pump in order to drive the conformational changes that occur when we pump out the calcium. So this is primary active transport. So the cell has various ways of maintaining this very low calcium ion concentration. And again, there are other ways, not just pumping the calcium out of the cell, but we can pump calcium into the mitochondrion. We can pump it into the endoplasmic reticulum. We can have calcium binding molecules in the cytosol that sequester calcium. So all of these mechanisms ensure that the calcium concentration in the cell, in the cytosol of the cell, I should say more specifically, is kept very low. So when the calcium ion concentration in the cytosol becomes high, that is a signaling event. So when we activate phospholipase C, I have another table right here, just like I had that big table for cyclic AMP. So when you activate phospholipase C, there is a series of events that results in the cytosolic calcium concentration going way up. So we activate phospholipase C by a G protein coupled receptor, the G sub Q, like I just talked about. But we're going to revisit this again. Excuse me. Receptor tyrosine kinases can also activate phospholipase C. So again, what happens when calcium goes up? Well, it depends on the hormone, it depends on the tissue, because there are all kinds of cellular responses that occur when the calcium concentration goes up. So let's talk about one of those. So this pathway, when you activate the phospholipase C beta, there is a phosphatidylinositol 4,5-bisphosphate, or we can just call it PIP2 for short, and that is present in small amounts in the cytosolic-facing leaflet of the plasma membrane. So when we've talked about plasma membranes, you know that we don't usually talk about phosphatidylinositol. It's usually PE or PC or PS and things like that. But we have PI in small amounts. So this is not drawn to scale. In fact, this is actually just showing the cytosolic leaflet. So we have a leaflet, say, above your computer screen, which faces the extracellular matrix, which is not shown. So they're just showing one leaflet of the membrane. So here's our phospholipid, here's our glycerol backbone, two tails, there's a phosphate, and this is called inositol. So this is this six-membered ring with lots of OH groups. So the cell has ways of synthesizing this PIP2. So we have to phosphorylate this at the number 4 position, and then we have another kinase that phosphorylates it at the number 5 position. So this is called PIP2 because this is a phosphatidylinositol 4,5-bisphosphate. So it's phosphorylated on the number 4 and the number 5 hydroxyl groups attached to the number 4 and 5 carbons. So basically, PIP2 just hangs out in the membrane and waits until phospholipase C beta is activated. And then when phospholipase C beta is activated, there is a hydrolysis of a bond that occurs right here. And what that does is it leaves diacylglycerol in the cytosol. So here's the glycerol backbone and the two fatty acids. So notice this isn't a phospholipid anymore because it's lost not only its head group, but its phosphate. And this is IP3. So it had those two phosphates on carbons 4 and 5, plus the phosphate that was part of the phospholipid. So this is IP3, very water soluble. Diacylglycerol is not. It happily stays in the membrane. But we have two second messengers now, and they do two different things. Diacylglycerol activates protein kinase C, and it's IP3. Its job to release calcium from the endoplasmic reticulum. So the pathway is splitting into two branches here. So here, again, this shows more of the chemistry, and this is in our textbook. So this is phospholipase C, and it's a hydrolysis right here. So again, if we do the hydrolysis right here, then we release the IP3, and then these R groups are just the fatty acids. So the diacylglycerol stays in the membrane, but the IP3 is very water soluble and can diffuse through the cytosol. So this is what happens. So here is the PIP2, and then the phospholipase C is shown in yellow. So we do this hydrolysis, and the diacylglycerol activates protein kinase C, and protein kinase C does what protein kinases do, it phosphorylates substrates. But these pathways come together a little bit too, because calcium ions bind to protein kinase C, and that activates it. And also there's a translocation of protein kinase C from the cytosol to the membrane, where diacylglycerol activates it. Also, protein kinase C is activated by phosphatidylserine, and you might remember that that's in the inner cytoplasmic leaflet. But anyway, back to IP3. That travels through the cell to the endoplasmic reticulum and binds to these IP3 gated calcium channels and opens the channels, and now calcium floods the cell. So protein kinase C is one of the proteins that the calcium ions activate, but there are other targets as well. So now what we've done is we've raised the concentration of the calcium in the cytosol. It goes from 10 to the minus 7 to 10 to the minus 3. So it's a huge increase in calcium ion concentration. So remember, we have to turn these signals off too. So when the signaling event is over, then what happens is IP3 is dephosphorylated to IP2, or it can be phosphorylated to IP4. So IP4 is another intercellular mediator, whereas IP2 is active. But that's a way where you can decrease the concentration of IP3. And because all of the calcium has entered the cytosol, now we have to pump it out. So all of those ATPases that I just showed you will pump calcium out of the cell entirely, or back into the mitochondria and back into the ER. Okay, so what about this diacylglycerol branch? So we can do another hydrolysis to cleave it to arachidonic acid. That's also a signaling molecule, or we can use it to synthesize eicosanoids, which we have signaling properties in the inflammatory response. And you might remember this from nutrition, because there are certain dietary ways that we can influence this pathway. Or it can activate protein kinase C, another serine threonine protein kinase, which was shown on the last slide. So let's talk about that. So here is a picture from our book, and again, this shows the activation of phospholipase C. So back at the beginning, the signal bound to the receptor activated the G protein, this is G sub Q, and then GDP was swapped for GTP, and we dissociated from gamma and beta, and then the target was the PIP2, and the PIP2 was the substrate for the activated phospholipase C beta, which was activated by the G protein, and then we do the hydrolysis. The diacylglycerol stays in the membrane, activates protein kinase C, and there again our phosphatidylserine that's just naturally present in the cytosolic leaflet of the cell that also activates it. And then this shows the IP3, which opens up the calcium channel. So the calcium can do a lot of things. So one of them is it can actually bind to a protein called calmodulin. And calmodulin can bind to other kinases and activate them. And then also we're showing up here, protein kinases are also phosphorylating their targets. So there are lots of kinases that are active in this signaling pathway that phosphorylate proteins and lead to a cellular response, and that response depends on the cell that you're in. So again, the concentration of calcium rises because IP3 opened those gates and allowed the calcium to come in from the ER. So that alters the protein kinase C conformation. It translocates from the cytosol to the cytoplasmic face, and then protein kinase C needs calcium, diacylglycerol, and phosphatidylserine. And then once it has all of those things to activate it, then protein kinase C goes around and phosphorylates various target proteins. So let's talk about calmodulin that was also in that picture that I showed you. So this is the structure of calmodulin, and so calmodulin is a calcium binding protein, and the calcium ions are shown here in cyan. So there's 1, 2, 3, 4 calcium ions, and then there are specific amino acids that ligand the calcium, and those are shown in sticks, and the rest of the molecule is shown in a ribbon form. So calmodulin is a single polypeptide chain, so it's got 4 high affinity binding sites. And when calcium binds, there's a pretty large conformational change that happens to the calmodulin. So let's look at that. So here is here the calciums are shown in yellow, and this is the calmodulin, and it has this hinge region right here, and this is the conformational change that occurs. So this is if we have this target protein right here in red, we can see the calcium calmodulin can bind at this hinge region and just completely wrap itself around this protein. So here the calmodulin is activating that target protein, whatever it is. So this shows biochemically a little bit what's going on, and it shows kind of this hinge region, and there are these hydrophobic patches that allow it to bind to certain hydrophobic patches of the protein. But the binding of the calcium causes this conformational change that allows the calmodulin to wrap around a target protein and then activates that protein, whatever that target is. So there's the cartoony picture right here, but I thought maybe you'd want to see more biochemically what was going on. So in this picture, the calcium again is shown in cyan, and the calmodulin is shown in blue and in red, and the target right here is shown in green. So once we have calcium calmodulin, then what happens? Well, there are lots of kinases that depend on calcium calmodulin. So one of them is myosin light chain kinase, and that's involved in muscle contraction. But the other one is phosphorylase kinase that I just talked about. So that was involved in glycogen breakdown. That was the phosphorylase kinase that was phosphorylated by protein kinase A, which then activated glycogen phosphorylase. So let's talk about that. So if we want to fully activate phosphorylase kinase, so again, so I called it, so this book calls it glycogen phosphorylase kinase. I just usually call it phosphorylase kinase. So remember, to put that in context, phosphorylase kinase activates glycogen phosphorylase by phosphorylating it, and that activates glycogen phosphorylase, and then glycogen phosphorylase catalyzes glycogen breakdown. So anyway, this is from my molecular biology book, so excuse the improperly labeled amino and carboxy termini. So if this is phosphorylase kinase, and if it's completely inactive, what happens is that this is very cartoony. What phosphorylase kinase really looks like is it is a multimer. So it has an alpha, beta, gamma, delta subunits, and there are 4 of them. So the gamma subunit is the catalytic domain, the alpha and beta subunits are phosphorylated, and the delta subunit is actually calmodulin. So here calmodulin is actually part of the protein, which is not really shown very well in this picture, but it does show how it's activated. So I have my protein phosphatase, and then when calcium binds to calmodulin, we get this conformational change that occurs, and then that activates the phosphorylase kinase. But phosphorylase kinase is also activated by phosphorylation by protein kinase A. So what this diagram is meaning to show is that the protein, the phosphorylase kinase, isn't completely active until it is both phosphorylated by protein kinase A and activated by calcium binding to calmodulin. So in the muscle, when we are exercising, we can have both neural and hormonal stimulation to make sure that our muscles are getting the energy that they need. So we've talked about before about how epinephrine can bind to receptors on the muscle cell, activate the G protein, activate adenylate cyclase to increase the concentration of cyclic AMP. That activates protein kinase A, and protein kinase A will phosphorylate glycogen synthase and uh, halt this pathway because you're exercising, you're not synthesizing glycogen. The protein kinase A also phosphorylates the glycogen phosphorylase kinase, which then phosphorylates glycogen phosphorylase, and then that means we have increased glycogen degradation. So this is what we want when we're exercising. But this neural stimulation will also increase the intracellular concentration of calcium. So we have both cyclic AMP and calcium that are collaborating to fully activate glycogen phosphorylase kinase, because when the calcium concentration goes up, that's going to activate that delta subunit, the calmodulin, and that will result in fully activated glycogen phosphorylase kinase. So we will really get glycogen breakdown, which is what we need to do when we are exercising. In the liver, okay, so that was in the muscle. In the liver, we still have hormonal stimulation, and we have collaboration of these pathways. So we have hormonal stimulation. So this is what you're most familiar with cyclic AMP activating activating protein kinase A, and then phosphorylating the synthase to make it inactive, and phosphorylating the phosphorylase kinase to make it active, and that glycogen phosphorylase to make it active to increase glycogen degradation. So this is what's happening in the liver during starvation. But we can add more players to this. So we also have the stimulation of the phospholipase C beta, which produces diacylglycerol and IP3, and the diacylglycerol activates protein kinase C. So glycogen synthase is a target for glycogen synthase as well. So both protein kinase C and protein kinase A are collaborating to make sure that glycogen synthase is phosphorylated and inactive. And protein kinase C, remember, needs calcium to be fully active. So over here with the IP3 causes calcium to be released from the endoplasmic reticulum, and that's going to help the glycogen phosphorylase kinase be active because of the calmodulin subunit, and that will ensure that the glycogen phosphorylase kinase is fully active so it can phosphorylate and activate glycogen phosphorylase, so glycogen is broken down. So by having the collaboration of these multiple signaling pathways, we can be absolutely sure that glycogen synthase will be phosphorylated and inactive, and we will not have glycogen synthase because during starvation we can't be doing that. And we will have increased glycogen degradation, which is good because that will provide glucose for the rest of the body during starvation. So I wanted to end with a couple fun slides. So a long time ago, I worked in a laboratory at Indiana University, and the lab next door to mine was run by Alexandra Newton, and she is an expert at protein kinase C, and she's not at Indiana anymore. She moved to University of San Diego, but she's a multi-talented person. Not only is she a great scientist, but she's also a great artist. So she made these drawings of this fish, and here's the receptor which activates the G protein. Here's phospholipase C, and PIP2 is shown as this little crab which activates the octopus and then phosphorylates its substrate, and there's IP3. So she made t-shirts with this. So If you remember the old SOA study lounge, that's the couch outside of my office. And there was no biochemistry 2 till the spring of 1996. And that was the first time that I got the course through Faculty Senate. And I had my first class of students. And they didn't have to take this class. It didn't count for anything, but they took the class anyway. So I had these students back in the spring of 1996, and I bought them all these t-shirts. And I am in touch with a lot of these students still. A couple of them are doctors. So we have a doctors, researchers, high school teachers, and an airline pilot among these students. Here's the airline pilot. He calls me my birthday every year because my birthday is the day after his mom, so he always remembers. He calls me every year. So I showed that picture to our friends, the class of um, 2019, and they took the class in 2018, and they decided they wanted a picture too. So this is a much higher quality picture. So again, there's the couch outside my office, and they said they needed a picture because they would be the last class to study in the student SOA lounge because of the renovation. So you look at that glass, that atrium, that is not there anymore. There is a cold room there. They wanted to be the last ones to study in this space. So we have a picture of them too. So we have ways of remembering all of the classes in between and to come later. So I hope you enjoyed the cell signaling lecture, and I will see you next time.